When I first saw this, it was like amazement. Here's a situation where this piece of flesh, essentially, I mean, really, it's a piece of flesh, will actually go on to restore missing body parts, such as the head. I mean, not just like a little appendage or something, the actual head grows back. Wow, that's remarkable. They just move so elegantly. They look like ice skaters, just gliding effortlessly. Makes them interesting. I mean, when I see a planarian, I just see a big void of mystery in the sense of how an animal that is relatively simple and so different from us could actually do things that for us would be very, very good to understand. Regeneration, this reshaping of pre-existing structures to achieve the appropriate form and function after amputation. And that they are doing this with genes that, for the most part, you and I have. Anything that you can surmise from studying these animals will have some effect in the way we think about our own genome and the way we actually function and why we look like we look and why we have organs like the ones we have and so forth. This is one day post-amputation, and there's this black area, this dense collection of pigmentation at one end, which is missing on the other end. So this is the tail tip of the animal. So that's what happens on day one, and the animals, as you can see, it's not moving very much. That's because it's lacking photoreceptors, so they cannot sense light. On day three, the animals may or may not move, but they're not going to respond to light in the same way as they would if they had photoreceptors. So all of this white tissue in front of the pigmented area, that is chock full of the daughter cells of the resident stem cells migrating to the wound to produce what's going to be the head. And so if you now go to day 6, you can see this much better. They are now responding to light. So probably the light is coming from behind, and so the animals are trying to move away from the light because they don't like light. By day 7, most of the regeneration, that is the differentiation and patterning of the head, has been completed. And I know that because I can look at the pigment cups right here, these 2 black dots that you can see now in this sea of unpigmented tissue. So the photoreceptors will only appear once the brain is really well formed. And now that brain will take over the rest of the old tissue, the pigmented structures right here, and begin to guide it and make the rest of the tissue behave like a planarian. This is a trunk undergoing regeneration, and I can pretty much tell already which one is going to be the head and which one is going to be the tail. And the reason for that is that here's the pharynx, and even though this is the back of the animal, not the belly, this right here would be where the pharynx comes out from the bottom of the animal. So that's my guess, that this is the opening of the pharynx, this is going to regenerate a tail, and this is going to regenerate a head. You can see again the little pigmented areas right there. So this is day 3. It's already beginning to move, but in no direction at all. Here's again the head. You can see the photoreceptor pigment cups right there. Here's the tail. This whitish area is where the pharynx resides when it's inside of the animal. And again, you can see very well the pigment cups in this head blastema. And here's the tail again. So this animal by day 7 is pretty ready. So here's an animal that doesn't have a head or a tail. So how does it know that the surface that was closer to the head should give rise to a head? And how does it know that the surface is close to the tail should give rise to a tail? In other words, how do they know their positional information? How is this stored? Given that the structures are missing and that the animal is regenerating both structures at the same time, how does the new head control the new tail? What are the rules of engagement here? Not clear at all. So this is another thing that planarians can do. Here's a situation where the animal has been amputated along the midline of the body axis. And what happens now? Well, each half will regenerate its mirror image. And so let's look at this movie. Here's the head. It was probably cut as well through the midline as possible. I can't see the photoreceptors on this particular specimen, but you can see the auricle right here. And I bet you anything what's going to happen now is that this tissue is going to fill in, and there's the new regeneration blastema right there. This white unpigmented area. In fact, you can begin to see the signs that an auricle is going to come up right around there. And then this is the rest of the animal. The pharynx is regenerating right there. See the white area right here? This is a beautiful amputation. This is very, very close to the midline. And you can now begin to see a better shape of the head right here. The animal is moving about. The pharynx has already begun to regenerate just fine. Now you can see it's controlling the rest of the new tissue already. That means that probably the brain is there, and there is the new photoreceptor right there. Oh, that's gorgeous. And then you can see the unpigmented structure of the animal. So it's regenerating along the midline quite beautifully. And the auricle, you can tell which one is the new one and which one is the old one by the lack of pigmentation. And at the earlier time point, you can actually see that the auricle was smaller than the pre-existing auricle. But by day 15, it's pretty much done. 15 days for regenerating half of your body, that's not too shabby. Given that we don't know what the basic fundamental biology of stem cells is in mammals or humans, maybe if we can do experiments quicker in planarians and come up with a set of principles that says this is what you need to keep a population of stem cells. What are those genes? Well, let's look at the human genome. Turns out the human genome has this gene. Has it ever been described to play a role in the stem cell biology? Well, nobody has looked. Let's look. Oh, look, it does. Or, oh, darn it, it doesn't. So go back to the drawing board until you find it. This is why working on model systems is so important, I think, because we can do many more experiments, test many more hypotheses, and the hope will be that the evolutionary conservation will emerge, will become self-evident, and you can transpose that information to humans. So I think we can learn a lot from not just looking at planarians, but almost everything around us about our own biology.